We concluded the last lecture with this slide that, based on quiz results and things, everybody seems to have a very good handle on overall. I'm not going to repeat it, but I just use this as the closing point of the fundamental steps of DNA replication. The material today that I'll cover talks about a couple of special cases, as well as how these things can go wrong with some of those warfare agents that I introduced the last class, as well as with other environmental agents that can disrupt DNA as it's being replicated. I wanted to start with one additional problem of DNA replication that's specifically to linear chromosomes, linear DNA. And that is what happens, based on everything that I told you in the preceding lecture is correct, what happens to the ends of that linear DNA as you replicate. In fact, what occurs is you have this thing called an end replication problem, which I can cartoon for you here. If we take this as the parent strand, primase lays down an RNA primer, right, that then gets elongated by Pol delta, says before. But the problem is that when you have a linear strand of DNA, a linear chromosome, at the end, there's no end over here. There's no DNA at the very end for Pol alpha. So I'm not saying 1 or alpha because 1 is prokaryote circular genome, so it doesn't have this type of problem. Right? So Pol alpha has no DNA to bind to be able to exert its 5' to 3' exonuclease activity to prune away this RNA primer. So it persists, except it's not stable. It's not stably bound to the DNA, and it's not covalently bound to the daughter strand. So it's going to get eroded. Hydrolysis, RNA is less stable than DNA. Right? It goes away, and it never gets elongated again. Now this is the parent. Another round of DNA replication, RNA primer, daughter strand. Now that one whittles away. Okay? And so this happens at a base by base level resolution at the ends of each one of our chromosomes. The ends of linear chromosomes are called telomeres. And what we have evolved to be able to resolve this end replication problem, or at least forestall it for as long as possible, is by using the function of an enzyme called telomerase. Telomerase is an enzyme. As you can see on the slide, it's an RNA-dependent DNA polymerase. And the DNA polymerase, it's still a DNA polymerase in the sense that it only can elongate an existing strand. But one of the special characteristics of it is that it is preloaded with an RNA template. So it is bound constitutively to a piece of RNA that acts as a primer. Telomerase will go and use that bound RNA-DNA hybrid here to begin elongation. It fills it in through the end of the RNA primer. And if the projection or your slide deck will permit, you'll know that it started by binding to a TTG. At the end of the elongation through, it creates another TTG at the end, creating another binding site for telomerase to bind there and elongate again. And so this TTG, you can see the sequence there, I'll read it off. That repeat occurs over and over again at telomeres. It's called a telomere repeat. And it's characteristic of the ends of linear chromosomes. And that's by virtue of the telomerase activity, keeping those ends from growing them back as they're progressively whittled away during each round of replication. As I mentioned, the telomerase can forestall the erosion of these ends, but it doesn't prevent it completely from happening. It doesn't block it. It offsets what's occurring as a result of not being able to bind to the very end of the DNA. And so in circumstances where telomerase expression is low, adult tissues, like that, if there is a big burst of proliferation and then an erosion of those telomeres, the cell will stop proliferating but not die, just sit there. Okay, so sit there, be metabolically active, but no longer able to proliferate because the ends have been whittled away, and it sets up a response called a senescence response. So senescence, old age, okay? And just sit there and kind of hang out, senior citizens of our cells. Yes, question?
So
Yeah, will it add to extragenic DNA? Yeah, telomeres are non-coding, definitely extragenic DNA. And you can kind of think of them almost like insulators from the process of DNA replication getting shorter and shorter at each round. They only apply to lagging strands, but over multiple rounds of DNA replication, they're both going to be the lagging strand, either in one round of synthesis or the other. But you're correct. It's only relevant for each strand on one side, but then they're the other side at the next round of replication. Yes?
You know how on skin, near the bottom, like underneath there, when the cells are proliferating and dividing stuff, but on the top, they just kind of like eventually grow through yourself. Would you say that the top layer of your skin is senescent, or that's still alive?
Oh, so your question about skin and senescence almost perfectly scoops what I was about to say. But the process of squamate differentiation and like the sloughing off of your skin, that's another thing called cornification that occurs. But where senescence immediately occurs, you see on the skin, most of us can see, are moles, nevi. All right, so when you have a little black mole and all of a sudden it grows for a little while and then stops, senescence. Okay, what happened was it actually had undergone a couple of mutations, lack of growth control, proliferated, grew for a while, but then the senescence kicks in and then it just stops and it won't proliferate again. That's why you go to the dermatologist and the most concern are not the fact you have moles, but the moles that are growing. Okay, either they've escaped a senescence block or they've found other mechanisms to evade that means for tumor suppression. Yes, so your question is where might telomerase come to bear on carcinogenesis, the formation of tumors? If you'll just grant me one moment. I hate being in the spotlight here, so I'm going to try to. That's manageable? It's much more manageable for me. I feel like I'm under interrogation. Yeah, so the question on where might telomerase come into to bear for cancers. I can say one thing is that if you take a fibroblast, like a ECM, I'm trying to use the vocabulary that I know you've taken in the class already. Yeah, so fibroblast ECM secreting cells in your skin, if you plate them out on a dish, they'll grow, a certain number of population doublings, depends upon the organism, but let's say they double 50 times, and then they just stop, senescence. If you take those fibroblasts and then drive overexpression of telomerase in those cells, you can what's called immortalize them, meaning that they'll replicate indefinitely, more or less. There are additional inciting events that need to occur for you to form a full-blown tumor, but it is something that can enable you to avoid replicative senescence. And so there are other means by which you can bypass this process also during tumorigenesis. And so the reason why I'm hesitating a little bit is for me to say that it's a real cancer-causing gene. I can't think of a tumor right now that has explicitly upregulated. There are other bypass mechanisms to avoid senescence that I know are more readily disrupted in cancers. And we're going to do another lecture on cancer at the end of the semester. Yeah?
So senescence basically when it starts cutting into the actual organism, and then at that point it's a non-viable organism and it stops replicating?
Yes, so when you talk about telomere erosion, once you have eroded the entire telomere away, there's no longer the telomere repeat to enable the extension with telomerase, even if it's there. And so that's going to create a signal, which in my last reading of the literature is still a little bit enigmatic about what precisely is. I could speculate, but my speculations will be meaningless until I get to the other slides to talk about what those signals might be. But if these exposed ends of DNA are pretty bad for the cells, and that is thought to be the trigger to turn on this senescence machinery, no longer proliferation, and off you go. Yeah, and so this a specific challenge overcome by telomerase in organisms with linear chromosomes. All right, so that's the last bit of the bits and pieces of DNA replication. We're going to go on a bit of an aside now to talk about anthrax, get back to anthrax. We're going to get back to mustard gas now that you've had a foray in the components of DNA replication. I spoke briefly on pathogenesis of inhaled anthrax. I'll speak a little bit more about that now. If it gets delivered via the lungs, the internalization of those anthrax spores occurs by lung macrophages that are trolling your lung surfaces, looking for bacteria, virus, any other things to phagocytose. Remember the professional phagocytes? And so if an anthrax spore lands in the airways, those airway macrophages internalize the spores, and then when that phagocytosed spore goes into a phagocyte of the macrophage, that creates an environment that's favorable for the spore to start proliferating again. So the change in pH and the other microenvironment of the spore cause it to germinate, it was like seeds, and then it begins proliferating inside the macrophage. It will continue to proliferate inside the macrophage until it lyses, and then all of those released bacteria can be internalized, and process repeats again with more macrophages in the lung where there are many. The motivation for talking about anthrax was really to give you a sense of what the pathogen is, because that's important for the way that its antimicrobial, Cipro, acts. So that's the first key distinction. Before we get there, I want to do a brief introduction on antibiotics and antimicrobials and the difference between the two. This is straight terminology that's good to be aware of for those aspiring medical people in the audience. Antibiotics, we've probably heard that more commonly. These are compounds that have activity against microorganisms, but that they are derived from other microorganisms. The one that you should be aware of is penicillin, the bread mold one, okay? Alexander Fleming, all that type of stuff, okay? Antibiotic. Antimicrobial, if antibiotics are this big, antimicrobials encompass antibiotics, but include a larger realm of things that synthetic chemists have made over the years, and other types of substances. So it's a broader term. And you'll see why the Cipro, when I show you the chemical structure, is an antimicrobial. A few more things about both antimicrobials and antibiotics. What are the pharmaceutical chemists and things looking for? For many of you might know, we have a shortage of antibiotics and an increasing problem with antibiotic resistance, antimicrobial resistance. What are the chemists looking for? You want toxicity, but it needs to be selective, meaning that you need to target a process that's in the microbe, and I'm using microbe generally to refer to bacteria and fungi, namely. So you wanted to act on that microbe, but not the host. There's some practical things, stable, easy to administer, as cheap as you can charge for it, I guess. The third point here is an important distinction that anybody who's interested in microbiology needs to get an appreciation for. Bactericidal versus bacteriostatic. If you can envision a population of anthrax, germinated anthrax bacteria, and then they're proliferating. If you put on a bacteriostatic agent, the growth will be, let's say, logarithmic for a while, and then just stops. So you stop the growth, but that's all you do. Bactericidal will take that population of germinated anthrax bugs and then eliminate it, okay? Cause death of those microbes and get the population down to zero. Bactericidal is essential, really, for good antimicrobials, because if you have a bacteriostatic agent, all you're doing is creating a breeding ground for antimicrobial resistance. If those cells are hanging out there and they're not getting eliminated, it's more time for them to pick up a plasmid, another piece of DNA from the environment. It's more time for those microbes to pick up a mutation that renders them resistant to your antimicrobial. And so bacteriostatic is not a desirable property. It can be confusing if you're looking at microbiology experiments, because usually the way you'll do those experiments is you'll start with a small inoculum of bacteria, right? And then your negative control, which is no intervention, it grows. So you say, oh, look, I went from here, and it's all the way back down to the starting level of the culture. Look at how effective it is. But if you don't get the level down below from what it started, you can't tell if you had a bactericidal or a bacteriostatic agent. And this is the reason why microbial people harp on this. And I already talked about that. It relates to resistance. One that you may have a sense of anecdotally, but whatever antimicrobial that is administered to a patient, it needs to get to the target tissue where the infection occurs. And different drugs, different antimicrobials or antibiotics, have different distribution profiles, meaning they accumulate in different parts of our body, and they have different excretion profiles, meaning how do they exit us. And depending upon the route that it goes in and the route that it goes out, and where it visits along the way, these are going to determine what are the right, this in part will determine what's the right antibiotic for the right infection. Adverse reactions, I think that goes without saying. I want to expand a bit more on this notion of spectrum of activity and what appropriate spectrum of activity. We can divide the spectrum of activity into broad and narrow. Basically, you can think of this as the number of microorganisms that any antimicrobial or antibiotic is effective against. Number of bugs that are susceptible to the agent. Narrow spectrum antibiotics will target a specific class of microbes. Maybe it's gram-negative bacteria. Maybe it's a specific class of gram-positive bacteria. Okay? It depends on the agent. Broad spectrum antibiotics, kind of broad swath, hit a lot of different things. And so you might think that, oh, why not just treat with broad spectrum antibiotics all the time? Sometimes you have to. You don't know what the infection is exactly, and so you have to do it out of necessity. But one of the major concern of use, and especially overuse of broad spectrum antibiotics, is that you can have unintended consequences as a result of just wiping out all of the microbes in a person. We have in our bodies pounds of commensal microorganisms in our guts and all our mucous membrane, air passages, things like this. And when you take one of these really wide spectrum antibiotics, if it has broad tissue distribution, you disrupt a lot of that. And so what can happen is that extensive use of these antibiotics can give rise to things called superinfection, where you wipe out the original bug of interest, but all the commensal microbes that ordinarily exist on a person, and that creates the opportunity for an invasive species to come in that's antibiotic or antimicrobial resistant and overtake the individual with lethal results. One of the examples that people might have heard is C. Clostridium difficile, C. diff infections in emergency rooms. This is a bug that can't really survive in our intestines under normal circumstances. It's only under individuals that are immune compromised or have been taking antibiotics, broad spectrum antibiotics for long periods of time, wipe out the commensal the bacteria, and then this C. diff bug can grow. It's resistant to these antibiotics, and that's where it's a leading cause of lethality in emergency rooms, intensive care units, long-term care, hospitals, and so on. The analogy, if you want to wrap around your head around this, would be an ecosystem argument. Right? If you you kill all the predators in an ecosystem, right? All the wolves and the eagles or other types of things. Then what happens is that the deer and the rabbits proliferate because they don't have anyone to keep them in check. And then the secondary consequence of that is all the vegetation disappears. Okay? And so there are these cascading effects of taking an ecosystem and doing these broad-scale perturbations have unintended consequences on the overall ecosystem itself. Left first.
You mentioned broad spectrum being used in immunocompromised individuals. What other applications?
One of the broad spectrum antibiotics, that you have some where if you have a really rapid onset infection and you don't have time to do a culture or a PCR, you don't know, you just rapidly intervene. And now I'll defer to the MSTP student in my lab. Are there any other places where the broad spectrum antibiotics?
They're used if you have a drug-resistant organism.
Okay, yeah, that was the other one I was going to say, that if you try the, let's say, frontline therapy, I think I'm misusing oncology terms, but the first thing that you would treat with, if you're not seeing the responses, you could say, well, I probably guessed wrong on the first one. Let's go with a more aggressive treatment, which is the broad spectrum.
So, does the broad spectrum, like wiping out all the or does that prevent microbes from directly interacting with the super infected bug, or does it prevent the microbes from creating an environment where that bug can survive?
yeah, okay, so the question, which I'm recording for the video, is that, well, why do you have that? Why would you take away one, open up all the other one? A lot of where the competition, there are examples of, let's say, a secreted factor from a commensal bacteria being deleterious on other microorganisms, but the overwhelming mechanism for keeping them in check is competition for nutrients. And so if you have an organism that's more fit in the natural environment, let's say it's in the gut, it can extract nutrients from that environment, grow faster, and just outcompete these slower growing but more antimicrobial resistant organisms. So it's just surely a numbers game of can they get the nutrients out of the environment and take them away from the other ones that don't grow as fast. Follow on?
So that super infection plug, that would be resistant to the broad spectrum, right? And that's why it's fine.
Correct. So it only in the selective pressure of that broad spectrum antibiotic can it then flourish. And there's actually some examples of this also in the cancer realm. Now, that's much more my comfort area than microbiology. But you have the same thing with anti-cancer resistance as well. You treat really heavily, you eliminate 99.99% of the tumor, but it's that 0.001% that survives. There are some colleagues of mine that I know that are doing things called adaptive therapy, where you go on and off chemotherapeutics to kind of keep you don't try to eradicate the tumor, you just try to shrink it a little bit and then let it grow a little bit so that the faster growers can outcompete the really nasty ones that you don't have any treatment for. You treat them again, it shrinks again. It grows a little bit, it shrinks again, it grows a little bit. And they're prolonging patients. There's some pretty remarkable results for pretty aggressive. They're using it for prostate cancer. So ecology coming up in a couple of different facets of molecular and cellular biology. All right, cool. No blue terms on this. All right, I just want to give you a feeling of the types of strategies for antimicrobials. And when I list them, they seem obvious, but it's good to see them all. The root of good antimicrobials, this is broadly, is to target fundamental bacterial or fungal processes that have specificity to the bacterial and fungal realms that wouldn't impact us. As an example, we don't have a cell wall. All right, so inhibitors of cell wall, that's what penicillin does, are, I believe it's among the earliest antimicrobials, antibiotics identified. You can also disrupt cell membranes. So fungi have cell membranes. They have specific requirements for how their cell membranes are assembled. You can develop inhibitors for that. Protein synthesis, well, you'd think by protein synthesis, that seems a bit odd because we need to synthesize our proteins as well. But there are differences in prokaryotic ribosomes compared to eukaryotic ribosomes that can be exploited to design prokaryotic specific inhibitors. Several are listed here. There are also differences that could be exploited for metabolism. I'm walking very gently here because we have a whole number of lectures coming up on metabolism. But there are key pathways to synthesize. You do know deoxyribonucleic acids, so purines, all right, A's and G's. Those are essential for DNA replication, proliferation, and there are means by which to inhibit the enzymes in bacteria that are required to synthesize those building blocks for DNA synthesis. And then there are also other ones, more broadly, facets related to nucleic acid function. This is where we're going to zoom in, and this is where Cipro comes up. Cipro, full name, ciprofloxacin. It's one of these quinolone antibiotics that I mentioned very briefly at the end of the last slide. Here's the chemical structure. You can tell by its simplicity. Maybe you think it's complicated, but if I showed you penicillin or any tree-derived compound, you would see that those are much more elaborate chemical structures. That's what's shown here. This comes up in a screen when chemists take simple compounds. They put a fluorine here, and then I'll put a carboxylate over here, and put this, I don't even know what this chemical structure is, see how that impacts the activity, and screen for function, right? And then this is what they get out. Very powerful. It's the only one that has shown some efficacy in anthrax, although the N for this is very small. And the reason why we brought it into this lecture is because its mechanism of action deals intimately with the gyrase in bacteria that is important for relieving the supercoiling that we talked about last time. And so if you have a rapidly proliferating population of anthrax bacteria, right, and then they're treated with Cipro, what will happen next? Circular genome, replication fork, opening, 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 opening. All right, but if gyrase is inhibited, you get this huge backup here, just like we showed in my demonstration last time, right? Backed up, backed up, backed up. And then at a certain point, the torsion and the supercoiling on that DNA becomes so extreme, the helicase, you can't go any further, right? But the helicase is going to keep trying, the polymerase is going to keep extending, and as you keep forcing the issue, all right, what will happen is the DNA will break. And so so-called stalling of replication forks, which is what happens here, so one way in which you can stall it, if sustained, will give rise to DNA double-stranded breaks. And it's this DNA damage as a result of being unable to relieve the supercoiling on the circular genome. It's the mechanism of action of Cipro in anthrax treatments. All right, so there's an application of DNA replication, pharmacology. Let's touch now on the chemical weapons world. As I mentioned in the beginning, mustard gas is a vesicant. I look every year, and I never find a satisfactory explanation of why that chemical agent gives rise to the blistering. I had a couple of hypotheses, but I've never been able to come up with a good primary paper to vet that out. So it's unknown still, and there's not too many people working on a World War I chemical agent. But what we do know is that it performs chemistry on a lot of biomolecules, and one of them is DNA. And specifically within the DNA, it modifies guanine. Mustard gas on the left, you saw this chemical compound before. On the right-hand side is a guanine base. What occurs, as I mentioned last time, this chloride here is a good leaving group in this in the mustard gas head here. And so what happens is that the amine here has some free electrons that are going to go and attack this carbon, release the chloride, and that happens 2 times, right? Because there are 2 of those good leaving groups. And now what you have is a covalent modification between 2 closely spaced DNA bases on the genome. These are very bad for replication in a number of different ways. The spacing of those 2 groups is such that you have enough reactivity to form cross-links, so GG cross-links. If you have 2 Gs next to one another on the same strand, you can form an intrastrand cross-link. You can, there's enough spacing on that molecule to go across the strands and form interstrand cross-links. And even if you don't hit 2 Gs in a row, even if you just put 1 on there, you've still put a big alkyl group on one of the chemical bases, on one of those guanines, that's going to disrupt the base pairing when replication occurs. And so if you have a polymerase here, it's going to stall at the cross-link. If you hit an alkylated group, that's also going to cause the stall because it can't find the appropriate base pairing. And then if helicase comes in and encounters one of these interstrand cross-links, great way for DNA damage to occur because it's going to keep plowing on through there, but it's chemically, covalently tied together, another mechanism for DNA damage. And so collectively, all of these chemical modifications are what gives rise to the profound DNA damage that occurs as a result of exposure to mustard gas. So stalled replication forks, and when you have stalled replication forks, this is how you'll get strand breaks. Interestingly, mustard gas or mustard gas derivatives have been used and are still used as chemotherapies. We already touched on cancer a little bit today. We can speak about it a little bit more. They don't use the mustard gas like I showed you before, but they will use these mustard heads, and I just updated this slide this morning because I wanted to show it to you. This is a chemotherapeutic called bendamustine. It's approved to treat chronic lymphocytic leukemia, multiple myeloma, and non-Hodgkin's lymphoma. You can see the mustard group here. Here are the 2 chlorides, the good leaving group. All right, it's an amine now instead of a sulfur. All right, but the premise still is the same. Cross-linking and alkylating DNA with these. Rationale, cancer cells are rapidly proliferating. They're going to have proportionately a higher time when they're replicating their DNA compared to normal cells. Still some nasty side effects with this, okay, because your proliferating cells are going to be affected. But the idea is that your non-Hodgkin's lymphoma or multiple myeloma is proliferating that much faster and will be that much more susceptible to the alkylating agent, intra-strand cross-links, stalled replication forks, DNA damage. We use this slide as the segue to talk about DNA damage more broadly, because DNA damage and the repair processes that cells have evolved to wrestle with that DNA damage are intimately tied to the responses to certain chemotherapeutics, as well as the response to certain carcinogens that we are exposed to in our environment. This slide comes in 3 parts. I'm going to give you a list of representative examples of DNA damage. They're not mutually exclusive. It's not uncommon for you to see mixtures of these depending upon the chemical agent. But the first one to touch on is straight removal of a base. All right, a purine. All right, so we have a guanine here, which is a purine. There are certain environmental agents. Oxidative stress can do this. Oxidants can hydrolyze this glycosidic bond and just take away the entire base. Right? And this is called depurination. When you depurinate a residue, now you have no template to fill in. Right? And so you can imagine how this could be a source of mutations in a genome. We can take away purines, which is to remove an entire base. We can also take away specific chemical groups on a base that changes the way that it's interpreted by the DNA replication machinery. And the classic example of this is deamination. So in this example, if you have a cytosine here, you see this amine group, it gets removed by a process of deamination, and then as a result of taking away that amine, there's a reconfiguration of the ring, and now all of a sudden it becomes uracil. Uracil, we think we touched on briefly in the ribonucleic world. Right? There's no reason it can't exist in a deoxyribonucleic acid through this chemical modification, but then during the base pairing, it's interpreted as a different template. It's going to look more like a TU, all right, pairing to an A than a C to G pairing. The one place where you've already seen cytidine deamination, although we didn't tell you that at the time, was during somatic hypermutation. Remember somatic hypermutation, Professor? Oh no, I told you that over video. All right, that rapid evolution of the FAB binding domains, hypervariable regions to increase the affinity. How does that occur? There's an enzyme called adenosine to inosine deaminase that takes those cytidines and removes that amine, and then puts point mutations all the way around in that locus. So the AID or AID is localized to the hypervariable regions, puts point mutations in there by modifying the DNA, and then that's selected for during B cell expansion. When you have when you take the either of these are going to cause one of these mutations occur or cause mismatching of the base pairs. And then the last one that I'll talk about is when we sit out in the sun. I think the last number I heard is that we accrue 75,000 of these per hour in the sun. And those are thymine dimers. Ultraviolet light has enough energy in it that if you have 2 adjacent thymines on a strand, you can form a chemical crosslink between those 2. Another form of DNA damage. Now, as I mentioned, we have tens of thousands of these events happening every time we're out in the sun. Right? The vast majority of them are fixed. Right? This is a spin the wheel, there's damage, it gets recognized and then gets repaired. So I hope this provides a motivation for why we have a deep need to understand the processes of DNA repair, which is what you're going to get the next couple slides. So you're stuck here with me regardless. The mechanism of DNA repair, I give you a laundry list there of different ways in which the DNA can be damaged. There are others that I'll talk about later on. But the first point to take home is that the mechanism of repair is a function of the damage that is trying to be repaired. The base excision repair machinery, BER machinery, is to handle these types of events, these single or stretches of individual nucleotides that have been damaged. I will not torture the group, but all of the individual names of the repair enzymes, we're going to give you the gestalt of it by saying what are the key steps. The key steps is, first, there's a recognition of the base damage by what are called repair endonucleases. We talked about endonucleases before. Here you see them in practice. They need to cut inside a DNA strand, not on one of the ends. And the thinking is that because you've depurinated a base or you've undergone a deamination event, now the base pairing there right at the site of the damage is no longer as nice and neat and as normal looking as if it would if it were a proper base pair. So you have kink in the helix that could in principle be recognized by these repair endonucleases. So you get recruitment binding to the site of DNA damage, and because they're endonucleases, they will cut the strand. And these kind of endonucleases will only cut one strand. Right, so they cut, excise there. This now creates a gap in the genome that will then get filled in by a polymerase. It's mostly polymerase. Hold on. I wanted to look at this. Yeah, depending on the size of the gap, if it's a longer gap, perhaps as you might imagine, polymerase delta can fill in. It looks just like a DNA replication event. If it's a single base fill-in, there's a specialized polymerase called beta that will do those, but that's getting into the weeds. But here, you have a polymerase, fills it in all the way up until it can go no further. Then you have that gap. It's filled in by a DNA ligase, and all done, repaired. Use the template here from the undamaged side where the base that was damaged. So you excise that base, thus base excision repair. Fill it in, paste it together, and off you go. Rather different, yes?
What would happen if both sides were
You set me up perfectly, because that was exactly what I was going to say next. Is that, well, what happens if you damage both sides together? All right, that's a totally different category of DNA damage. And how does that happen? It doesn't happen with lightning bolts, okay? But it will happen with ionizing radiation. It will happen with stalled replication forks. It will happen with any agent that causes a stalled replication fork, chemotherapeutics and things. Like cosmic rays. I don't know, am I forgetting anything else that would cause double-stranded breaks? You could do it. We're going to talk about ways in which you do this intentionally in a slide or two. But the major ones I think of are stalled replication forks, chemotherapeutics that cause stalled replication forks, and ionizing radiation. Ionizing radiation. You blast the genome. By the energy of the radiation, you break the DNA in two. As I think you were alluding to, you don't have the luxury now of a template to draw from in the way that it's currently schematized. And so the path to double-strand DNA break repair has a fork in it and can go in one of two paths. I'll tell you about the simpler and the less elegant version first, and then the more complicated one on the left-hand side. The simpler and less elegant one is called non-homologous end joining, or NHEJ. NHEJ is a brute force means of getting that double-strand break back together. The general strategy is when it gets broken, it's oftentimes not a clean break. There are exonucleases in the nucleus that can start doing a little bit of whittling away there. What occurs is that when you have the two ends, there will be some filling in and pruning away the two free ends to try to make them compatible again to get pasted back together. So you may have removal of some bases, insertion of some bases, and then once you have enough complementarity to be able for them to get ligated back together, it gets ligated again. Junctional diversity, has that already lost your memory banks? Remember the imprecise joining of V's and D's and D's and J's? Okay. Why do you get those insertions and deletions? It's NHEJ. Right, so you have the break from VDJ recombination, and then there's an attempt to try to get those two to be compatible and join them back in together. if this double-strand break occurs in an extragenic region, there was a question about extragenic regions before, this works great most of the time, right? Because you don't have any genes there or anything. It usually works fine. If you add a couple bases or take a couple bases away, it's probably not the end of the world. So it's a great machinery because it's active all the time and really effective in cells. This can be a lot more problematic, though, if that double-strand break occurs in a coding region.
Oh, extragenic, that would be like a non-coding area, right?
Yeah, yes, outside of genes. Intronic would probably be the same within the same thing. But where the real difference, the real danger runs is when the double-strand break occurs in a coding region, in an exon. All right, that codes for the DNA. Now, walking delicately on this, every time you have a break, all right, 2/3 of the time, when you add and take away individual bases, you're going to disrupt the protein expression of that. And if you'll just let me say that, and then there'll be a lot more explanation when we talk about protein translation. But often when you start adding and removing bases, you're going to disrupt the way that that gene is translated and the way that it's expressed. So over evolutionary time, there's an alternative pathway that's a lot more complicated, but a lot more elegant. We're diploid organisms. We have 2 copies of every gene. Let's say one of them gets broken, double strand. There's a different form of pruning that occurs on the 2 ends where the double strand is called end resection. But you notice that the overhangs are different. The goal is not to try to paste them together. The goal is to try to get enough whittled away so that there can be called a homology search. These ends here will diffuse around, look in the nucleus, to find something that looks like a reasonable complement to that. How about the other strand of DNA, the other chromosome, the other region of the gene? If it's close enough, this also happens a lot when the cell's already doubled its genome and there's twice as many copies to go around. If there's a complementary copy of the gene, this end will invade into the other DNA, use it as a template to fill in the gap, and then bridge back over to the original strand that it started on. So the gist of it is that it's using another highly similar piece of DNA that's in the nucleus at the same time as a template to fill in the lost information that was a result of the double strand break and that end resection. Concept of that. There's a lot more details in this and things, but I'm sparing you that. All right, because what I want to do is build upon these 2 processes of NHEJ. Oh, sorry, I forgot to tell you what that is. This is called homologous recombination. And where the homologous part comes from is it's looking for a homologous copy of what is missing and using that as a template. And the recombination is because it's moving a piece. The net result is to take a piece or the information from another piece of DNA and encode it into the place that got repaired. So the colors here you pay attention to because where did the information come from to fill in the gap? These 2 DNA repair processes come to a head when we talk about applications of CRISPR Cas9, the extent to which question. Yep. It's important to know this first before we talk about the next thing, because then it will be lost. So.
Is that broken strand, how does it find its complementary strand? Is it just floating around and then it bumps into it?
Yeah. The question is, how does it? Yeah, it strikes me as remarkable that it ever finds that complementary strand. A couple things to say on that. One is that if you are we haven't done cell cycle. Okay, if you've not doubled your DNA, all right, if you're just sitting there and not actively growing, very little homologous recombination happens. It's a fraction of a % of the time. Even when you have doubled your genome and you're about to divide, when you have 4 copies instead of 2 copies, even then it only works effectively maybe 10% of the time, and it depends upon the gene locus and things. We actually were just doing a paper in my lab journal club on this, where they would try to do this in a bunch of different chromosomes, and the range is, I don't know, it was like 10 to even up to 50% but depending on the region that you were targeting, it could vary widely. And my suspicion is that some of it has to do with the proximity of the two chromosomes in the nucleus. There's quite a bit of stereotype structure of where chromosomes reside inside the nucleus. Therefore, if you had two alleles of the chromosome just happen to be right next to one another in topologic space in the nucleus, I would suspect that those would be better candidates for HR-mediated repair. Whereas if they're all over the place in the nucleus, it'd probably be just pure luck, diffusive luck. Mm-hmm.
Why would these cells be like in the dividing context? Would the damage initiate cell division or something like that?
Oh, why would you get damage when you're dividing? Well, we have some tissues in our body that are always dividing. So if I blast your gut, there will be some cells that are dividing, right? So just turn, your hair follicles and things like that. Over long evolutionary time, cosmic rays and all these other types of things. So cells are always dividing, and so there's an advantage to being able to have an additional fail-safe mechanism to have a better fidelity of what's been inherited up to this point. That's, I think, is the rationale. There was something. Yes? Oh, okay. Yeah, so you cut, or there's a break, and now you have these two ends. Why don't they just like peel open? You've seen cartoons of like, dynamic instability with microtubules and things like that. Yeah, the base pairing, the thermodynamic stability of the base pair is strong. I think we talked about DNA melting in the pre-lecture about how you can increase the temperature and then you can cause the strands to fall apart, and then you drop the temperature and they come back together. Therefore, thermodynamically, at body temperature or ambient temperature, it's favorable for the two DNA strands to stick together.
I'm sorry, I meant not like the purple versus the blue, but when you have the cut.
Oh, these two go all over the place.
They do.
Yeah. But what happens is now you have two free ends, no telomeres. Okay, so those free ends that don't have any, that's a trigger for recruitment of proteins. There's a big complex that gets formed. There's an assembly that occurs. I'm not going to give you all the names of those, but you recruit a whole bunch of molecules to the break site, and those binding proteins will concentrate there. I think that they will keep them spatially kind of nearby to do this. But as you can see, like for homologous recombination, the two ends don't, there's a search process first, and then you have binding proteins on either side, which will come together and do the final joining.
And I guess in neither case it will ever be a clean break, because if it was a clean break, that they would just thermodynamically reanneal, right?
Okay, so if you took it and I just like snip on the two sides, would they come back together? In that circumstance, no. Because let's just say even if you had like one or two bases, perfect overhang, and it could be complementary, if you calculate the melting temperature of a 1 or 2 base pairing, it's not enough to keep it together. So on the length of a chromosome, right, when you have all this, the stretch, those won't fall apart like this, the way that I misinterpreted the question from before. But if you're talking about bringing like these two fingers together and it's only a base pair or two holding them, not enough thermodynamic favorability to overwhelm thermal fluctuations. Okay. So this is my last slide, so I just want to the rate at which I go through this. How many people have heard of CRISPR Cas9 in the news or things like that? Okay, how many people have not? Be honest. Thank you. Okay. Yeah, and so I'll start from the beginning. So CRISPR and Cas9 is a system. It's a system of, probably the simplest way to describe it is it's a type of adaptive immune system that some prokaryotes, so bacteria, and some in a totally different phylum, is that right? Yeah, I think it's phylum, called Archaea. These are the microorganisms that live in deep sea vents and the salt marshes and all these other types of things, have evolved to have a memory of the viruses that they've encountered over their evolutionary lifetimes, and as a means to combat these, to be able to mount a response to phage viral infections over time. that's the reason why it exists in evolution. All I'm going to do in the 6 minutes that I have is talk to you about how this CRISPR system, Cas9 and Cas9 enzyme, have been repurposed by biotechnologists, molecular biologists to enable what I guess we're calling genome engineering, using the capabilities of this enzyme and the natural processes of double-strand break repair to insert or remove pieces of genomes in any organism that I think has been tested up to this point. Have I hyped it enough for you? All right. What is Cas9? It is an enzyme. It is an endonuclease. This is the bubble diagram here. It has a few important characteristics. First is that it has 2 endonuclease domains. The white bubbles there, don't worry about the acronyms underneath them. But in addition to being an endonuclease with 2 separate endonuclease domains, it also is an RNA binding protein. And the RNA that it normally binds, in fact, binds 2. One of the ones that it binds is called a tracer RNA, and then a CRISPR RNA, which is the red, and then this other piece here. The CRISPR RNA is the part that's stably inherited in the bacterial or archaeal genome. Part of it base pairs with the tracer RNA. The other part contains information about phage viruses that had been infected in the bacteria in the past. For biotechnological applications, we learned that you could simply hook these 2 together with a hairpin here. So make one long RNA, a little loop here, another loop, and then go here. And this is called a single guide RNA, or an sgRNA. So in the wild, it's tracer RNAs and CRISPR RNAs. When we use it in the lab, it's sgRNAs. And that's just for convenience. When Cas9 is loaded up with the appropriate RNAs, it does a form of homology search, if I can repurpose that term. And it will look in the genome of the organism for a region that is complementary to this part of the guide RNA in red. There are some requirements about what that guide RNA needs to look like. That's what the protospacer and this protospacer adjacent motif, the PAM motif. But if you have a region of sequence there that is recognized in a DNA genome, and it has one of these PAMs nearby, the endonuclease activities of Cas9 are active, and it cuts both strands of the DNA wherever it bound and annealed based upon its targeting sequence, as shown here. That's the remarkable characteristic of Cas9, the ability to localize an endonuclease activity, localize an enzymatic activity anywhere in a genome based upon the RNA that it's loaded up with. Have I made that point clear? Because it says, oh, it's amazing. Why? Nothing else. There are other Cas9-like enzymes, but that was the thing that's so remarkable about this, and that's why this will win the Nobel Prize imminently. How do we use it in the lab? Well, we just talked about these repair processes before that happen naturally. Now we have a means recombinantly, okay, biotechnologically, we can put a double-strand break wherever we want, just based upon what we load up Cas9 with. When you have that double-strand break, you can do a couple of different things. If we want to knock out a gene, Dan talked about knockouts, right? We don't, the way that, when that paper that he cited, the way they do knockouts, nobody does knockouts that way anymore. We all do it this way. And the way that you do it is you take Cas9 and you target it. You've done exons and introns, at least in an introductory sense. We'll target Cas9 right to the first exon of a gene that we want to knock out and put a cut in it. The double cut doesn't itself break the gene and knock it out. What does is the non-homologous end joining here, that inserting and removal of a couple of base pairs, as I mentioned, 2/3 of the time, is going to disrupt the ability of that protein to form. So we don't remove the gene entirely. We simply remove the ability of that gene to encode protein, which, if you're interested in protein function, is good enough. So that's how we make knockouts these days. The second part, which is harder for all the reasons and the challenges with homologous recombination that we talked about before, you can also use this to knock in genes Dan also talked about knock-ins, the same slide a couple of weeks ago, by, in addition to driving expression of Cas9 and a guide RNA in the cells, if you give that cell also a homologous looking piece of DNA, that's this part here. So this is a piece of DNA that was added into the cells that has homology here and has homology here. It has the homology arms. It looks like a great template for homologous recombination. We usually add it in way many more copies than 2n or 4n. And what we're trying to trick the cell to do is use it as a template for homology-mediated repair. And now let's say this little green section is, it's green, so let's say it's a green fluorescent protein. It's a fluorescent label that we want to put fused to a gene of interest, which may be in red. So the way that we do that is you cut right at the place where you will preserve the translation of the protein that you're cutting. It reads from here. Now it reads into your green fluorescent protein, and then it goes and reads through the rest of the gene. People do this in the lab all the time now. This was virtually impossible to do as of 10 years ago, 5, 10 years ago. But now people do it pretty routinely. And so this ability to take things away and put things in at will is one of the things that's been really empowering about the genome engineering revolution enabled by this class of enzymes. So if you think these things are neat, I'll talk about a retired faculty, Joyce Hamlin, who was the chair of biochemistry and molecular genetics a number of years ago. And I'll also give a shout out to a ninja who is the current chair of biochemistry and molecular genetics, both with interests in DNA replication. Be good for the TAs on Thursday. I'll see you on Tuesday.